So we're going to now talk about the next part of the pathway. So we're leaving the eye and we're going into the brain so we can complete the pathway. And we last time, like I said, we talked about the ganglion cells. And so we are now going to go to what it's connected to, which is the LGN, or the lateral geniculate nucleus, most of them. So let me go ahead and do this. So just remember, the whole point of doing this is so that we can look at this particular image and say to ourselves, I'm going to be confident that I understand it, I know what's in it, and I understand kind of what we're dealing with when I look at this. So, again, here's our standard OCT that I've always shown you. I don't want you to be nervous. Remember that the retinal circuitry is complicated. It's not completely understood. You want to repeat it from different angles like we're doing. So we were here where we said, okay, photoreceptors, bipolar cells, ganglion cells, we finished this. And so now we're actually moving into the brain. And here's your brain, sort of from the ventral surface. Okay, there's your LGN. That's the thing that we focus on, the lateral geniculate nucleus. I believe this is from your book, but I'm not sure. But it's similar enough to other things in your book.
Yeah.
So here's your lateral geniculate nucleus. We're still in the main neuronal pathway. I just want to point out one thing. This is from 2001, which is a long time ago, but not like 1957, okay? And here's a line here. The lateral geniculate nucleus is a huge embarrassment to visual neuroscientists, okay? I just want you to realize that we are entering now a spot, and we will come back to the retina and also have a similar kind of feeling, but we are entering a spot where we had made more and more discoveries like recently, like, in the last like 20 years. I mean, I'm guessing you and I had gone, or we're like somewhere in college or about to go to college, whatever, but like, it's not like it was before our births or something. Like, things were being discovered like legitimately in our lifetime. And one thing in particular that will make you be like, oh yeah, that's a big deal. So let's go through and say, this is normally how they say to you. They say, okay, ganglion cell, nerve fiber, it's LGN, like ganglion cell to LGN. Right? I mean, that's the next thing, right? So this is normally how they were drawn. Okay, great. No, but we know it's not that. We know it goes through the nerve fiber layer. This is the axon of the ganglion cell, okay? Through the optic nerve, okay? Decussates at the chiasm, goes through the optic tract, and then goes to the lateral geniculate nucleus. So there's a lot of things that go on in order to get to the LGN. Here is where the LGN is. Now, this is you can see the skull spinning here, but what is that red thing? That red thing on the brainstem, or sort of like coating the brainstem? That's the thalamus. Okay, so where is the LGN when we look at it? Well, here's our thalamus. This is a sagittal section, okay? There's anterior to posterior, okay? And then we look, and here's another one where we have anterior to posterior, but now you can see that the lateral geniculate body is actually sitting on near this ventral surface, but like near basically the back side of the thalamus. And so the pulvinar is basically like the backside cheek, so to speak, of the thalamus, right? So you have that, and then these two little things kind of sit out of it. One is a little bit more lateral, okay, and a little bit more anterior, but a little bit more lateral. That's the lateral geniculate body. So let's look at it from a different angle. This is your coronal section. We're cutting through the brain from a different angle. And now you see here, it's actually very, very kind of interesting when you cut it at this layer, what the LGN looks like, right? It's very striated. So I zoomed in there. That's kind of what it looks like, kind of blurry, but let's make it less blurry. And here's what in this case, well, I mean, I'll just say the one side of the lateral geniculate looks like, and nucleus looks like. And so here we're looking at what we name, and you're already seeing there some of the words that we were using to describe the pathways. Here we see magno, and there we see parvo already. So if we schematically draw it out, now I've added a few other things here. I've shown you there's the M's, which correspond to magno. There's the P's that correspond to parvo. And then you get these K's in between. So you're like, okay, well, here's some K's, but I don't see some K's over there. So what we say to ourselves is, okay, well, let's draw them in over here. Oh, and one thing I'll point out is, if we look at the layers, before we do that, if we look at the layers, okay, again, here are the Ks. If we look at the layers, we say there's 1, 4, and 6. Layers 1, 4, and 6 receive input from the contralateral eye. Now, remember, this is post-chiasmatic. We're already past the chiasm. So before, you were in the retina, and then in order to get to the lateral geniculate body, you got to go through the chiasm. So decussation has occurred, and now we're kind of split everything up. So we've got these decussations. We've got to figure out how to organize them. And so here you see 1, 4, and 6 are the contralateral layers. They're taking an input from the other eye, whereas, of course, 2, 3, and 5 are the ipsilateral information from the ipsilateral eye. So if we do, so you can see that right there. So I just want you to keep that in mind. That's very important. 1, 4, and 6 are the contralateral eye. 2, 3, and 5 are the ipsilateral eye. Does that make sense?
Yep.
Okay. So now we're going to look here and figure out why we're talking about parvo and magno, right? What does magno mean? It means large. So the reason why this pathway was defined as such is because when they went to the LGN, which is actually where they discovered kind of the pathway and were defining things, magno means large. Look at the larger kind of cells. The larger cells in this layer are in 1 and 2, so they called them magnocellular layers, or the M cells, they'll call them. And then, of course, parvo means smaller in Greek. So you say, okay, that's what parvocellular means. Well, look, the smaller cells are over here. And so they realized that 3, 4, 5, and 6 had small cells in the primate brain, and then 1 and 2 had big cells. And they called it a day for like 40 years. And then they discovered another type of ganglion cell called a K-type bistratified ganglion cell, which we talked about, what I drew in yellow. And then they said, well, where do they go? And they followed them out. And somewhere around 2000, I think, I'm guessing in that time, they realized, wait a minute, they're here where we thought nothing actually was there in those white lines, those white spaces that you can see here, the white spaces. They thought that they weren't really, no synapsing was occurring there. Turns out they were. They were actually tiny, tiny cells, which are dust-like cells. So thus they named them conio because they're smaller than parvo. Okay, that's what conio means in Greek, dust. Okay, so now you get why they're called magnocellular, parvocellular, and coniocellular pathways. I have no doubt that had we discovered these guys way back when we discovered these, it would be like small, medium, and large, right? I mean, like that's what we would name them. But because we had magno and parvo, and then we found this guy, we're like, well, what's smaller than small? Dust. Okay, that's why they're called coniocellular. Okay, so now you know where they are. Okay, so let's talk about our classes. You remember the P-type midget ganglion cell, the M-type parasol cell. We talked about these are the ganglion cells that are in the retina. We're back in the retina. Here's the bistratified K-type cell. Then you've got your IPRGCs, which is the intrinsically photosensitive retinal ganglion cells that have melanopsin, so thus they can respond to light on their own. And the example I gave of it was a giant retinal ganglion cell. And then you have the other. Now, mind you, these things are the dendrites. This is going into the inner plexiform layer. All that stuff still exists. But let's go and now figure out what they look like when they go into the brain. So let's go to LGN. So we're in the ganglion cells. Now we're no longer ganglion cells. We're now following each one of these axons out and taking a trip with them. So let's talk about the midget P-type ganglion cell. Where does he go in the LGN? You already know, but we're going to talk about it. It's P-type. We also know it's called beta. I told you that before, and X. You don't have to worry about that, but beta. It goes via the parvocellular pathway. Therefore, they're touching what are the parvocellular cells, so to speak, or the parvo cells, or the P cells. They're also called the P cells in the LGN. Now, mind you, the P cells are neurons. They're not like, you know, little bits of stuff. Like one of the other residents was saying, oh, well, there's nothing going on there. I'm like, no, they're literally neurons, and that is a synapse, okay? So, you know, it's a synapse, just like you'd have a bipolar cell synapsing with the ganglion, right? You get what I'm saying? It's the same synapse.
Yep.
So, sorry, I'm getting feedback, Nitin.
I don't know, is that any better?
This is actually better. I don't know what happened. So actually better. So P cells in the LGN are neurons. They're neurons that go somewhere else in the brain. But just letting you know, you're synapsing midget P-type ganglion cell. Its axon will synapse with the P cells in the LGN. And we know this is responsible for red-green color vision. So here's your picture of them again. Here's the midget P-type cell. And we say to ourselves, great, okay, I can see that. Now let's go ahead and play around with this. If I drew my midget cell over here and I had his axon terminate in 5, layer 5, remember, it's only going to terminate in the parvocellular layers because it's a P-type cell. Well, this is going to come from, of course, it's 2, 3, or 5. This is going to be a ganglion cell that originated on the ipsilateral eye. Does that make sense?
Yep, yep.
And then if we, of course, go to 6, I know 1, 4, and 6. Well, that's the contralateral one. Make sense?
Yeah, okay, yeah.
Okay. Now let's do the parasol cell. It's easy, but you just have to remember the rules. That's all. So parasol cells, you already know, it's umbrella, it's dendritic tree, right? We're not talking about it's axonal. It's not like it's a parasol when it's in the LGN. That's the axon. We're talking about the dendrite when it's in the IPL, the inner plexiform layer. That's what defines a parasol cell. It's an M type or alpha. M type means the magnocellular pathway. Now you know it's called that. It's the M cells or the magnocells in the LGN, and it's low contrast motion for M, motion, right? Here we go again. Here's your parasol, the dendrites within the retina. You already know I'm talking about the retina because you see that there's a difference in the dendritic trees. But let's go ahead and go back to our LGN and say, here's one. Now I know because my schematic shows me that this is a nice, dendritic tree that looks parasol-like, and it's going here. It's got to go one or two. It doesn't have a choice, right? We already told you that it's a magnocellular cell. It's an M cell, or M type. Sorry, it's an M type ganglion cell. M type means it can only go one or two. It shouldn't be synapsing way up here. Then it wouldn't be M type. So it's going to synapse on one or two. Well, we know 1, 4, and 6 are contralateral. So this one's a contralateral. It's coming from the other eye. The axon is actually connected to a cell in the other eye. Does that make sense?
Yeah.
Okay, let's do the same thing. It's pretty simple. Here's 2. Okay, well, 2, 3, and 5 means what? 2, 3, and 5 means ipsilateral. Well, in magnocellular space, there's only 1 or 2, and 2, therefore, is ipsilateral. So we know it's going to be this cell, the axon, if I followed it back, literally will be in the ipsilateral eye. We now know that. Okay, great. Let's go to the bistratified K-type. This is, easy, I guess. K-type, we already know. What does K means? Coniocellular. Coniocellular means the dust cells. Now we know why it's called that. These are the K cells in the LGN, or the coniocells, so to speak, the dust cells in the LGN. We know it's due, the blue, it's always on to blue and off to red, green. It's done. It's also involved with color perception, like proprioception and integration, blah, blah, blah. Okay. And I drew this as a bistratified thing where here's the axon. So these are the dendrites. Okay, this is a bad image, but I'm trying to give you a representative image. Okay, so that's a K-type bistratified ganglion cell. Okay, now I'm giving you, because it's not entirely clear, like, how do you number these? There's a lot of white space all around. We could have had, this could have been a white space that was relevant. So we say, okay, where is 1, 2, 3, where is that? So we say, okay, well, here is K1, which is kind of, if you want to say, ventral or inferior, using ophthalmology terms, but it's like ventral. You remember ventral and dorsal. So it's like ventral to it and sort of medial to the corresponding, colored layer, so to speak here. So the magnocellular layer, the parvocellular layer is the colored layer. So K1 would be where? It would be right ventral or inferior or medial to that layer. So I'm going to draw them in here. Here are the legitimate layers. And for the most part, we're going to argue that they conform to the 2, 3, 5, 1, 4, 6 rule, okay, for contralateral and ipsilateral, okay? So let's go and draw them here and say, okay, we get rid of our, crutch of a cartoon there. And we say, okay, here's our bistratified cell and ganglion cell. And now where's this axon? Okay, well, it's there. Well, where is that? Okay, well, I see that layer 5 is here. And so this is underneath it. It's ventral and medial. That's how my number is. I start with 1 here. So this must be K5. If it's K5, then what does that mean? It's ipsilateral. So it's an ipsilateral eye ganglion cell that's bistratified. Make sense?
Yep.
And we do the same thing, right? I go find another one. Here's one here. It's 6, right? Okay, well, we know 1, 4, and 6 are, of course, contralateral. So this is going to be a contralateral bistratified K-type ganglion cell. Make sense?
Yeah, totally.
Just to say it over again, not to belabor the point, this has nothing to do with, like, he's a contralateral. Doesn't mean he's not a bistratified ganglion cell. He's in the K layer here. He's in the coniocellular layer. He's synapsing with a dust cell neuron. Well, that means that he is a bistratified, for the most part. Again, we may discover something in the future, but for the most part, okay, we know for a fact then he must be blue and bistratified. If I follow him all the way back to the contralateral eye, I'll follow him. I should see 2 stratification in both the on and the off sublamina, so to speak, sublamina A and B. Make sense?
Mm-hmm, mm-hmm.
Good. So great. Let's go to the IP RGCs. Giant, right? Well, where is he? He's melanopsin. I already told you. Ambient conditions, can respond on his own. Where does he? synapse, suprachiasmatic nucleus, etc., ambient light, circadian rhythm, okay? So that is not the LGN. So let's look at where the suprachiasmatic nucleus is. I don't necessarily wasn't thinking about this, but here is where it is. I'm drawing it for you. Notice, here is where the LGN would be, where the thalamus is, that's where it would be. But where's the suprachiasmatic nucleus? It's supra to the chiasm. I don't know why that never occurred to me before, but it's right above the chiasm, but it's not the LGN. So these are not synapsing in the LGN, they're synapsing somewhere else. Well, what does this deal with? I said ambient conditions. Here's olivary pretectal nucleus. This is another one. Here's another one that that these IP-RGCs respond to. Look familiar? This is the rostral midbrain. You can see the pretectal olivary nucleus. What do you see nearby that's like having like some sort of influence? The Ettinger-Westphal nucleus. What is that responsible for? Your parasympathetic innervation with your pupillary light responses, right? You remember this? So these guys are dealing with that. Makes complete sense. It's consistent with what their function is. When they're intrinsically photosensitive, it makes sense that they would be the kind of, like kind of side army that deals with this stuff while everything else can fail, I still can work. So it's kind of helpful in that way. And then, of course, the ventrolateral preoptic nucleus. I'm just throwing out some other places it is. Where's the VLPO? Here's your suprachiasmatic nucleus, there's your VLPO. So it's in the same spot, but it's not the LGN, okay? This guy deals with sleep, so circadian rhythms. So the point being is these guys are dealing, these particular retinal ganglion cells, and their canonical kind of classic, what we're using as a stand-in for them structurally is that giant retinal ganglion cell. These guys deal with sleep and all these things. They're not dealing with your standard visual kind of pathway. Just remember that. Okay, now let's look at the other. Here's the other, superior colliculus. I told you this before, superior colliculus, 1S and 2Cs, saccades, that's what they deal with. Where is the superior nucleus? Well, now we're looking again at this sort of the dorsal surface of the brainstem and the posterior surface of the brain here. You see the pulvinar over here, you see your lateral geniculate nucleus, and you see your superior colliculus, which is part of the quadrigeminal plate. You remember all this, right? I mean, you've heard of this before, yes?
Yep, absolutely.
Right? And so the superior colliculus is superior to the inferior colliculus, right? And notice something, they kind of draw it like it's going through the lateral geniculate nucleus, sort of, and then kind of synapsing on the superior colliculus. I'm not sure if that's actually true, but just know that that might be part of it, but they are not synapsing, even if they do go through the LGN with any LGN cells that we know of right now. So they control basically saccadic motion. Make sense?
Yeah, yeah.
Cool. So other. Good, and there's your pulvinar again, just to give you the sense of where you are. Okay, so once we, let's say, go back to our LGN cells, which we were doing with our standard pathway, our midget, parasol, and bistratified ganglion cells, they make up the vast majority of everything, right? 99% of our ganglion cells are those, 80% of which are midget, but 99% are those cells. They're going and they're synapsing here. After we go through the LGN, we transfer the baton to these M, P, and K cells. They are neurons. Where do they go? Well, they're going to go to the primary visual cortex, or V1. In particular, where do they go in V1? V1 isn't just V1. It's part of the cortex, but it's layer 4 of V1. This is something, unfortunately, you just have to know, okay? Layer 4 of V1, that's where we are. I mean, and you're an ophthalmologist, so like this is important because like this is where our vision is processed in the brain first. That's the first handoff in the actual way, in the visual cortex. So in the primary visual cortex, which is V1, notice layer 4 is not layer IV, okay? It's not V1 is the same as IV. V1 is just visual cortex first, and then layer 4 is layer 4 of V1. Make sense?
Yeah, totally.
Okay. And where is that in the brain? Well, it's in the calcarine sulcus. So let's go and see where that is. Here's a sagittal, specimen here, and you see where the calcarine sulcus is, right? And if we draw this out and we say, okay, let's get it. I may be from your book, may not be. Here is where you call your primary visual cortex. Now, mind you, I just want to mention something. Area 17 is Brodmann's area, right? The area 17 corresponds with the visual cortex. That does not mean it's V17. V1 is area 17 in Brodmann's nomenclature. Make sense?
Got it. Yeah.
Okay. And then we are in layer 4 is the first synapse from the LGN to V1. Make sense?
Okay. Yep.
Okay, so it's layer, the first handoff. It's like I'm driving to the city. I may be going to, New York City, but I'm going to Port Authority first. Okay, so it's like just because I'm in Port Authority doesn't mean I'm in the Port of San Francisco. Okay, I'm in the Port Authority and it is part of New York City. So layer 4 is like Port Authority. That's the first hand up, or Ellis Island. I don't know what you mind, whatever. Like it's where you first arrive, and then we spread out from there through V1. Then we go to V2, V blah, blah, blah. But like that's part of V1, which also his other name is area 17 of Brodmann's sort of notation, nomenclature. Make sense?
Got it. Yep.
Okay. So, and again, posterior brain occiput, right? That's where it is. Okay. So I am now going to do something that, when we started these, you'd think was ludicrous because it wouldn't feel like, you could do this, but now you can do this. So I'm going to point, to the LGN somewhere. You're going to tell me what ganglion cell, where it comes from, which eye, and what it does. And if you can tell which eye. So sometimes you can't tell which eye, right? I didn't specify for a few of the classes because there is no specification. But let's just say you can, then tell me. And then you tell me what its function is, okay? So I'm going to point to, there, okay? Go.
So we're in layer 5, which is the parvocellular layer, which means, so 2, 3, 5, it means any cell that came there came from so first of all, it's the P-type cell, because they're the ones that go in the parvocellular pathway, type midget cells, and it is coming from the ipsilateral eye.
It's coming from the ipsilateral eye. It's a midget. And then tell me what type of information is it carrying?
This is like slow conduction, color vision.
Got red, green, right?
Yep.
You got it. So P-type, beta, midget, retinal ganglion cell, ipsilateral, color. Perfect.
So this is also P-type, midget, contralateral eye.
Yep.
Same function as before.
And what is the function? Just say it.
Yep. Red, green, slow conduction. Yep.
Good.
Color.
Yep. It's always good to repeat it when it's early. So you got it. Okay. So what about that one? It's this white layer here.
So it's the white layer. There's the white layer behind 2.
Yep.
Layer we're saying. So that's one of the M-type cells. So that's
back up here. Back up here. Is it the M-type? I'm pointing to the white.
Oh, so the white, sorry, is K-type. It's dust.
Good.
It's not actually one of the, yeah. So it's a K-type. That's the cellular pathway. It comes from a bistratified cell from the ipsilateral eye, and it is responsible for blue, yellow vision, proprioception.
Good. Now, my question, when you say bistratified cell, specify what type of cell are we talking about?
It's a K-type.
But what type? Is it a bipolar cell?
No, K-type ganglion cell.
Ganglion. It's a bistratified K-type ganglion cell, right? So just remember that, because again, I'm asking for precision, not because I'm being pedantic, but because we know, because you've gone through these lectures, the bipolar cells have giant bistratified bipolar cells too. So if you say bistratified cell, we may not know. So it's perfect, but that way, and notice how like we can get to this level of granularity now, right? It's kind of crazy, right? Okay, let me go ahead and point. What is that? I mean, you see what it's called, but what is it called? Just repeat what you see there. Where am I pointing?
It's not the Edinger-Westphal, but it's just the thalamus.
Well, no, this is not the thalamus, right? So this is the pretectal olivary nucleus. Side, right? Not the thalamus, very important. But give me the type of ganglion cell that would synapse here.
So for the pretectal olivary nucleus.
I'll give you a hint. Well, tell me, these guys, you can see on this other side, he connects to the Edinger-Westphal. Edinger-Westphal deals with what?
Oh, yeah, which is the photosensitive ganglion cells.
Exactly, and intrinsically photosensitive ganglion cells. Yes. Tell me what it has inside of it that makes it intrinsically photosensitive.
Melanopsin.
Melanopsin. Tell me a few things that it deals with.
Like ambient conditions, circadian rhythms.
And pupillary response, right?
Yes.
You got it. Melanopsin, pupils, and ambient light. You got it. Okay. What about that one?
So this is the superior colliculus, quadrigeminal plate. And this is coming from these are like that other category of
Yep.
ganglion cells that basically project to the midbrain.
Yep.
And responsible was like saccades.
You got it. Okay, that one.
So layer four. So this is a P-type ganglion cell from the contralateral eye that deals with like 80% of ganglion cells. So slow conduction, color vision, red, green.
Got it. Now just tell me, if I were to look at this ganglion cell's dendrite, what would you describe it as? How would it look?
Small. They're small.
Good. Midget, right? You got it. Perfect. How about this one, this dark one?
So this is pointing to dark layer 1. So then that means it's an M-type magnocellular pathway, M-type ganglion cell, which is responsible for motion, fast motion, and it's layer 1, so it's coming from the contralateral eye.
You got it. Contralateral eye, parasol is what it would be. Contralateral eye, perfect. How about that one? It's the
white layer below 1. So this is a K-type ganglion cell in the coniocellular pathway. It's a bistratified ganglion cell coming from the contralateral eye, responsible for blue-yellow vision, appropriate vision.
Perfect. Perfect. How about that one?
It's a so that's layer 3. So it's a P-type ganglion cell, midget ganglion cell, responsible for color vision, red, green, coming from the ipsilateral eye.
You got it. Perfect. One more.
And this is the blue, the dark layer of 2. Yeah.
Yep.
So this is an M-type ganglion cell, this parasol cell, as you were mentioning, coming from the ipsilateral eye, responsible for fast motion.
You got it. And so parasol cell, ganglion cell, fast motion. You got it. Perfect. Okay. Now let's just do one little thing before we end. I'm going to lay this out here. Okay. You've seen this before, but let's follow, our nasal retina, which would correspond to the temporal visual field. We follow that, and that's the one that decusates, right? So we know that that's the one that crosses. So from either eye, it's the nasal retina / nasal temporal kind of visual field fibers, so to speak, that are decusating. Okay. All right. So we know that that's the one that switches. Now let's go ahead and look at a cross section of a LGN here, and we say to ourselves, okay, well, they're giving me this cross section here, so let me just, for fun, let's label this, right? I'm going to look and see, first of all, what is going on there. This actually is an arrow that they're showing here. I'm showing you this here. Like, what is this, right? So let's play with it. So we say, okay, well, let's start numbering, because that's how we started. That's how you've been taught, and I've been taught. How do we analyze an LGN? Okay, well, there's a dark layer there, so I'm going to say that's 1. Okay, and then I'm going to say that's 2. Okay, so those are our magnocellular layers. And then let's turn our heads here and say, okay, that's 3 if we go with it, and just assume that this kind of cuts across here. So we say 4, 5, and 6. Okay, if we go with that, then we know 1, 2, 3, 4, 5, 6. Here's 1 and 2. This is going to be magnocellular. 3, 4, 5, and 6. This is going to be parvocellular. And then we're going to say, okay, 1, 4, and 6 are contralateral, except 4 seems to be split in half.
Yeah.
What is this? So think about it. And I said to you that the stuff that's contralateral tends to be from which side? The temporal visual field, right? Yes. And thus that's the nasal kind of retina. And we know that it must be something that's going on near the macula, because we know that this is the parvocellular pathway, which is the midget ganglion cell, which deals with cones and color vision, which tends to sit near in the macula somewhere. So this is something is going on near the macula that is in the temporal visual field, or the nasal macula, that does not have any connection here, something in our visual field. What is this?
It's the optic nerve.
Correct. That's that's layer 4, we said, physiologic blind spot. Isn't that cool?
Oh, wow, that's really cool. Yeah.
See, now and you're like, I can do that by looking at the LGN. I mean, this is what I'm saying about our field is that it can do this, right? So, we're going to soon go to the brain and it's just like, wow, like we have the capacity because our things are static, right? And we can go from one thing to one cone. There are pathways that do that all the way to a spot in your brain, all the way to a spot in the LGN. So this is one of the cool parts about it.
That's
Okay.
really cool. Yeah.
So I will contact you again for the next one and hopefully this is helpful.
It's great. Thank you so much.
Awesome, man. Okay. Bye.
Take care. Yeah. Bye.